Hello everyone, good morning. Learning is not the product of teaching. Learning is the product of the activity of learners. COVID-19 pandemic situation has changed the learning method. Now, e-learning undertaken remotely through digital platforms. Here, Pulla College of Pharmacy brings GPRCP Hyderabad YouTube channel for digital learning through e-faculty development program, e-faculty student development programs, etc. Myself, Sheikh Nasib Basha, Assistant Professor, Department of Pharmaceutics, GPRCP, welcomes you Day 5 of 1 week e-faculty development program on research updates in pharmaceutical formulation and analytical method development organized by Department of Pharmaceutics and Pharmaceutical Analysis, Pulla College of Pharmacy in association with Indian Pharmaceutical Association, Telangana State Branch. We know bioavailability, bioequivalence, in vitro, in vivo, and their correlation, that is in vitro, in vivo correlation, etc., are the most important studies in formulation development. Further to concepts of bioequivalence applicability to different types of products is very important in formulation development. Dear participant, we have dedicated question answer session at the end of the talk. Please post your questions in the chat box. To discuss all these bioavailability, bioequivalence studies, in vitro, in vivo correlation, today we have Dr. Vijay Kumar Nagabandi, Deputy Manager, Formulation, Research Development, New Heat Pharma Technologies, Private Limited, Hyderabad. I am very glad to introduce today's speaker, Dr. Vijay Kumar Nagabandi sir. Dr. Vijay Kumar Nagabandi has completed his B.Pharm from Kakatiya University in 2004 with gold medal for securing university second rank. He had qualified in GATE 2015 with 97.6 percentile and completed his masters in pharmaceutics from Usma University in 2007. Later, he has worked as assistant professor for more than 8 years in academic institutions affiliated to Kakatiya University and parallelly completed his PhD in pharmaceutics from JNTU, Hyderabad. He believes in continuous learning and with his passion as a part of updating his knowledge. Recently, he completed PG Diploma in Patents Law from Nalsar University with university rank. He has received the 50 lakhs project grant from BIRAC, that is Biotechnology Industry Research Assistant Council, under SBIRI, Small Business Innovation Research Initiative, as key investigator for one of his industrial new molecule development projects. He has two patents to his credit entitled Process for the Preparation of Anthraquinone Topical Formulation and Stable Ready-to-Use Injectable Liquid Composition of levothyroxine or its salt and its process of preparation. He has published more than 30 papers in reputed national and international peer-reviewed journals. He is the author of two textbooks titled Experimental Approaches to Biopharmaceutics and Pharmacokinetics and Novel Techniques to Enhance Bioavailability of Poorly Water-Soluble Drugs. He has guided more than 30 students for their dissertation work at post-graduation level and 10 students at undergraduation level. He is the receiver of the Indian Pharmaceutical Association Sri Raman Bhai B. Patel Foundation IRF International Travel Grant from IPA Indian Pharmaceutical Association for presenting his research paper at American Association of Pharmaceutical Scientists Annual Meeting and Exposition 2012 APPS AM 2012 held at Chicago, USA. He has attended and presented his research outcomes at various national and international conferences. He is the life member of the various organizations like APPS, IPA, IPGA, APTI, IACP, ISPAR, and FICCP. Currently, he is working as Deputy Manager, Formulation Research Development, New Heat Pharma Technologies Private Limited since last 5 years. His research is mainly focused on formulation development of complex generics, and he is expertise in applying various bio-relevant tools in predicting bioequivalence results and designing of dosage forms without patent infringement, etc. Without further delay, I request Dr. N. Vijay Kumar sir to proceed for today's talk on the topic Role of in vitro in vivo correlation in bioequivalence studies and Bioequivalence. Welcome, sir.
Yeah, well,
Welcome,
thank you
Vijay
very
Kumar.
much, Nasib. Yeah, thank you very much, Nasib Basha, for such a nice introduction about me. I mean, you made it very easier for me to introduce myself. I mean, I think without introducing myself again, I can proceed for the presentation today. A very good
Thank
morning
you
to
very
all
much.
of you.
Please proceed.
A very good morning. A very good morning to all of you. I'm very sorry when I'm taking the webinar lecture on Sunday also. I mean, because when they have approached me, as I am working in an industry, it could not be possible for me to take the lecture on working days. So I requested them to arrange it on Sunday. So anyway, most of you might have accepted this and participating in the lecture. Before proceeding to the lecture, I would like to start the seminar or webinar with the prayer. It's a common practice for me, as my gurus taught me to start anything with common prayer. Guru Brahma, Guru Vishnu, Guru Devo, Maheshwara, Guru Sakshat Parabrahma, Tasmai Sri Guruve Namaha. Is very big thank to all the gurus in my life. Especially, I'm thankful to my guru and friend, my boss and my managing director, Dr. V. Venkateshwarlu Garu, for accepting this, accepting my request to deliver a lecture during this faculty development program, 1 week program on recent updates in formulation development and as well as analytical method development. And I thank you very much all the management team and especially principal sir and Dr. Prashanti Nasib Basha from Pulla Reddy College of Pharmacy for giving me this opportunity and for keeping me some belief on me. Definitely, I will try my level best to share my knowledge in role of in vitro in vivo correlation in bioequivalence and as well as in biowaiver. As you all know, even Nasib Basha also just now we said it is very important to have the knowledge on bioequivalence and to apply the IVIVC tools to get the bio success and also on knowledge on biowaiver. It is very important tool, especially in the formulation development. So most of the audience today, targeted audience, are faculty members from various academic institutions. I don't think that detailed explanation about IVIVC and the bioequivalence is not required. But today's seminar, I would like to focus more on case studies, how we can build the correlation between in vitro and in vivo, and how we can get the bio waiver, under what circumstances IVIVC is required, and under what circumstances in vivo bioequivalent testing is required, and when we can exempt the bioequivalence study, and how can we get the more success in bioequivalence studies. That I'm going to focus more with one case study in bioequivalence and one case study in biowaiver. So, as you all know, any pharmaceutical research, any pharmaceutical formulation research is generally, we have two types of researches. One is innovative research and other one is generic research. Innovative research, as you know, it requires at least preclinical studies, phase 1 clinical trials, then phase 2 clinical trials, then followed by phase 3 large number of patients. Then only the molecule can it can bring the molecule into the market as a suitable dosage form. So it requires more than 10 years of time. Sometimes it may go up to 20 years of time, and it requires the investment of more than 10,000 crores. So it's not a small job. And definitely the innovator product as a NDA, new drug application form, they will come into the market. They have some royalty, and definitely they will have some patents. But as a pharmaceutical or as a therapeutical alternative to the innovator molecule, there is another kind of research. Most of the Indian companies are focusing more on the other kind of research, is nothing but generic formulation research. The major difference between generic formulation research and innovative research is, as already the dosage form is existed in the market, as already clinical efficacy and safety has been proven, no need to conduct all the preclinical trials, no need to conduct all the phase 1, phase 2, phase 3 trials, because already safety and efficacy have been proven. Only the objective of generic research is to bring the copycat of existing innovator molecule into the market at low cost, because innovative pharma company have done so much research, elaborative research, by conducting preclinical phase 1, phase 2, phase 3 clinical trials. Even after bringing the molecule, phase 4 will be continued as post-marketing surveillance. So that requires more time, that requires more money. So that obviously the cost of the innovative product, it is very, very, very high and cannot be affordable by all the middle class people or below middle class people. So even government also will give the permission to do the generic research, because royalty will be given, monopoly will be given to the innovator for certain period of time. But behind that, if we bring the generic molecule, which is copycat or theoretical alternative to the innovator, then definitely, because all the studies are not required, the cost can be expected to be very less. So now the difference between innovator and the generic, innovator have done all the clinical trials, and generic no need to do all the clinical trials. But at the same time, all the clinical trials may not be required. But to prove generic formulation is equivalent to the innovator, certain criteria is required. The major criteria in the list of various requirements is the bioequivalence. At least we need to prove that our test formulation is bioequivalent to that of innovator formulation. So then only we can, even regulatory authority will give the approval to bring the molecule into the regulatory market. To bring the molecule into the market. So if the product is chemically equivalent, if the product is pharmaceutically equivalent, if the product is bioequivalent to the innovator, then doctor can alternatively prescribes our generic formulation as a theoretical alternative to the innovator molecule. So as the clinical trials are not required, only bioequivalent test is sufficient enough, the cost can be expected to be low. The generic formulation cost is expected to be low. So this is all known to all of you. So that's why I'm not going in detail about the bioequivalence and its importance. But any pharma company, if you consider 50% or 60% of weightage, or more than 60% of weightage from my side, I can tell you 60% of weightage can be given to the bioequivalence, because to bring the molecule generic formulation, to bring the generic formulation into the market, the major part, the major success story will be on bioequivalence part, because formulation development anybody can do. We can match the dissolution profile, we can match the other quality attributes as that of innovator very quickly and very easily by conducting several experiments. In vitro mapping is very easy. So we can do several experiments and several manipulations in the excipients concentrations and excipient ratios or the matrix. We can do anything, and we can map the dissolution profile in the lab, in the in vitro conditions. But once the moment when we conduct the bioequivalence testing, it is very critical to match the dissolution, to match the bioavailability as that of innovator. So most of the pharma companies will fail at the final stage while conducting the bioequivalence studies. So that is the reason why, if we have a tool, a biorelevant tool, which connects the in vitro dissolution or in vitro other testing parameter with that of in vivo parameter, which builds the correlation between in vitro and in vivo, and using that tool we can predict the bioequivalence before conducting the bioequivalence study, so that whether to go with the bioequivalence with that formulation or any other changes required to be incorporated into the formulation, that can be assessed by conducting the in vitro in vivo correlation, so that at least number of bioequivalence studies can be reduced, so that ultimately the cost of the formulation further it can be reduced down. That is the major objective of this today's presentation. I have chosen this topic because it is more important, and for especially the people who are working in the pharmaceutical industry and the faculty members who are working in academic who are teaching post-graduation students with pharmaceutics background, and while advising them to do their projects work, if they apply these tools, definitely it would be helpful for them once if they join back to their industrial, once they start their career in the industry. So that is the reason why I've chosen this topic. This is more important, and quickly I will finish the introduction part, and then I will move on to the IVIVC section, and I will finish with one case study in the IVIVC with respect to bioequivalence, in vivo bioequivalence, and one case study with respect to the bio-waiver. Here the bio-waiver is nothing but waiver is nothing but giving some exemption for conducting the bioequivalence. Bio in the sense bioavailability study, waiver is nothing but exemption. BE, I have given the title BE. BE is nothing but bioequivalence. IVIVC is nothing but in vitro in vivo correlation. So let me begin with the brief definitions first, then I will move on to the other important requirements, when the bioequivalence studies are required, when the bioequivalence studies are not required. If bioequivalence study is required, do we need to do in vivo study or in vitro study? If in vitro study is required, what kind of in vitro studies are sufficient enough to get the bioequivalence? All these topics will be covered in the introduction. Then I will move on to the IVIVC. So here you can see, just I have given the image in vitro. I have represented dissolution apparatus. Dissolution apparatus is in vitro, because most important quality attribute in vitro is the dissolution. It has more relevant to the in vivo performance, in vivo within the body. Upon administration, how the dosage form will behaves. As you all know, any dosage form, it is having similar API and similar excipients as that of innovator. It may shows same behavior in vitro conditions, but it may not behave similarly as that of innovator and in vivo, because in vivo it has so many other parameters will come into the picture, like GIPH and gastrointestinal enzymes presence and microbial flora presence, any efflux transporters and blood flow rate. So many other factors will come into the picture in vivo conditions, even body weight, physiological conditions in the GIT. All other factors will influence the dosage form. So though our test formulation behaves similarly in vitro, it may not behave similarly in vivo. In vivo performance may alter. So that may reflect in the bioequivalence testing. So that's why many formulations will pass in vitro testing, but it may fail in vivo bioequivalence testing. So if we have the correlation between in vitro and in vivo, it becomes easier for a formulation scientist to predict the bioequivalence. In our prediction itself, if it is going beyond the limit, then definitely that formulation is no need to test for the bioequivalence. We can do certain manipulations in the formulation, and again we can come back to the IVIVC. If that in that repeated IVIVC, if it is giving a good predictable result, then only it can be subjected for the bioequivalence testing. So this IVIVC tool, it will not give you any waiver for the bioequivalence, but it will give you the predictability. It will give you it will works as a tool to predict your bioequivalence result. And sometimes this IVIVC alone, it will helps you to get the bio waiver also. Those are all factors I am going to explain in subsequent slides. So here there are 3 levels of IVIVC, as you know. Those are 3 levels of IVIVC also I will explain in the subsequent slides. But let me begin with the definitions first. Bioavailability. I mean, just for having a quick recollecting of the definition, just I have given the definitions here, as if though you have already known about the bioavailability definition and bioequivalence. Just unless we recollect the definitions, it is very difficult to proceed further. Bioavailability, as you know, it is the rate and extent of the drug that enters into the systemic circulation. How much amount of the drug that is available at the systemic circulation, and at what rate it is reaching the systemic circulation, and of course ultimately at the site of action. That is the bioavailability. Bioavailability in the biological system, how much amount is available and at what rate it is available in the systemic circulation is known as bioavailability. And the important other definition is bioequivalence. Bioequivalence is nothing but when the test product is our generic product, when the test product bioavailability is equivalent to the innovator's bioavailability. Innovator product is reference product, our product is test product. Test product bioavailability, if it is equivalent to the innovator's bioavailability, then these 2 formulations are said to be bioequivalent. Equivalent. That means bio means bioavailability is equal. But there is a criteria before testing the bioequivalence. Our test formulation, chemically it should be equivalent, and pharmaceutically it should be equivalent. Chemically it should be equivalent means the API using in our test formulation, it should be equivalent to the API used in the reference formulation. API in the sense it is active pharmaceutical ingredient. It should be equal in the equal dose also. API should be, if innovator formulation is paracetamol, test formulation also should contain the paracetamol. And if innovator formulation is 500 mg, test formulation also should have 500 mg. This is nothing but the chemical equivalence. If innovator is diclofenac sodium, test formulation also should be diclofenac sodium. If innovator is diclofenac potassium, test formulation also it should be diclofenac potassium. That means even salt form also should be same, and dose also should be same. When the drug and dose both are equal, then these 2 formulations are said to be chemically equivalent. When they are chemically equivalent, the next part is pharmaceutical equivalence. Pharmaceutical equivalence nothing but if innovator formulation is tablet, your formulation also should be a tablet. If innovator formulation is a capsule, your formulation also should be a capsule. And if innovator formulation is administered through oral route, your formulation also should be administered through oral route. That means pharmaceutical equivalence nothing but it should be same dosage form, administered through the same route of administration, but we can have the liberty to use the different excipients. The drug should be same, dosage form should be same, but we can use the different excipients, different packing configuration, but it should mimic the behavior of innovator formulation in vitro conditions. Like it should have the same critical quality attributes as that of innovator. That means innovator formulation, if it is a tablet, your formulation also should be tablet. If it is oral route, your also should be oral route. their dissolution profile, your dissolution profile, their disintegration time, your disintegration time, their assay, your assay, their RS, your RS, their identification, your identification. All these critical quality attributes should be same. In that case, these two formulations are said to be pharmaceutically equivalent. So, provided your formulation is chemically equivalent, your formulation is pharmaceutically equivalent, then you are allowed to do the bioequivalence testing. Chemical equivalence should be proven, pharmaceutical equivalence should be proven, then only you are allowed to conduct the bioequivalence testing. And once you are proven that your formulation is bioequivalent to the innovator, then ultimately your formulation is chemically equivalent, pharmaceutical equivalent, bioequivalent, then absolutely these two formulations are therapeutically equivalent, because having the same drug, administering through the same route, and having the same dose, same dosage form, having same bioavailability, and ultimately the rate and amount of drug that reaches the site of action from these two dosage forms will be same. And when the drug is same, amount is same, rate is same, definitely we can expect same therapeutic efficacy from these two dosage forms. And in that case, either doctor can prescribe the innovator formulation or the test formulation. But as the test formulation cost is very less, I mean, because it is generic formulation, patient can choose, because it is easily affordable, you can choose either innovator or generic formulation. But obviously generic formulation has more benefits compared to the innovator because cost point of view. So that is the major objective of generic formulation development. But as said, copycat is very difficult job. Mimicking the same as that of innovator formulation is very difficult job. Here the bioequivalence, similar bioequivalence is nothing but, as you know, Cmax value and AUC value. These two are the critical parameters. Cmax is nothing but maximum plasma concentration achieved by the innovator formulation, and test formulation should be same. And AUC, area under the curve, achieved by the innovator formulation and test formulation, both should be same. Because as I told you, rate and extent of the drug that enters into the systemic circulation is known as bioavailability. Rate and extent, these are the two parameters, rate and extent. Rate is nothing but the Cmax will give you the rate of absorption, and AUC will give you the extent of absorption. Rate of absorption, extent of absorption. For rate of absorption, Cmax, extent of absorption, AUC. These two parameters are very important parameters in establishing the bioequivalence. Cmax should be same, AUC should be same. Same, nothing but 100% is not at all possible. Even if you repeat the bioequivalence with innovator formulation versus innovator, it is not possible to get the 100%. But here there is a criteria, 80 to 125 percentage. If your formulation Cmax value in between 80 to 125 percentage as that of innovator, your AUC value in between 80 to 125 percentage of innovator formulation. 80 to 125 percentage is the limit acceptance criteria given by the regulatory authorities. So if your formulation is in between 80 to 125 percentage, it can be said that your formulation is equivalent to the innovator formulation. Then, as already proven that chemical equivalent, pharmaceutical equivalent, and bioequivalent, you will get the approval as a generic formulation, and your formulation can bring into the market. So patient can take either innovator or the generic formulation, except that all other parameters are same, excipients can be different. So in this case, chemical equivalence proving is easy, pharmaceutical equivalence is proving is easy, but proving the bioequivalence is very tough job for any formulation scientist, because in vivo performance of any dosage form is unpredictable. So many other parameters needs to be considered while developing the formulation, where IVIVC role will play very key role. I mean, IVIC will play a key role in establishing bioequivalence and predicting the bioequivalence, because in vitro we are correlating with in vivo performance. So that is very important job that I am going to explain. So this slide will give you an idea when bioequivalence studies are necessary and what are the types of studies are required. You know, all dosage forms, all generic formulations may not require bioequivalence testing. There are some certain exemptions and there are certain limitations where the bioequivalence study is mandatory. You may do anything. You may do IVIVC tooling or anything. Definitely in vivo bioequivalence testing is required. But there are few dosage forms, even without IVIVC also, bioequivalence testing is not required. So those things I am covering in the next two slides. In vivo studies are required. These are the list of dosage forms where in vivo bioequivalence testing is required. That bioequivalence, either it may be sometimes pharmacokinetic bioequivalence, clinical pharmacodynamic study, or comparative clinical trial study. In the following categories of drugs, following categories of dosage forms, this is required. Example, starting with A, oral immediate release drug formulations with systemic action. when one or more of the following criteria applies. Any oral immediate release formulation which is indicated for serious conditions requires assured therapeutic response. Indicated for serious conditions, definitely in vivo bioequivalence testing is required. And any drug having narrow therapeutic window or narrow safety margin, steep dose response curve. This is nothing but high potent drugs. High potent low dose drugs, definitely in vivo testing is required. And pharmacokinetics complicated by variable incomplete absorption or variable absorption window, non-linear pharmacokinetics, pre-systemic elimination, first pass metabolism. These kind of drugs are also should be tested for in vivo bioequivalence testing. There is no exemption, there is no bio waiver. And any formulation having unfavorable physicochemical properties. If the drug is having low solubility, if the drug is having instability in the GIT, if the drug is having polymorphic behavior in the GIT, if the drug is having poor permeability, low soluble, low permeable, variable dissolution profile, instability in the GIT, and which is having the behavior to undergo polymorphic changes in the GIT, definitely we need to do in vivo bioequivalence testing. There is no chance of bio waiver. And if there is any documented evidence that bioavailability problem is there, that may be related to the drug, may be related to the similar chemical structure or formulations, in that case also bioavailability in vivo testing is required. And where there is a high ratio of excipients to the active ingredient, API to the excipient ratio, if it is very high, then also that means drug is very low, excipient quantity is very high. So even you have proven that you are chemically equivalent, but the excipients quantity is more, how these excipients going to impact the in vivo performance, we don't know. That's why in vivo testing is required. And in continuation, that is one category of oral immediate release dosage forms, and other category non-oral and non-parenteral formulations. Like if any formulation is administered through transdermal route as a transdermal patch, suppositories, these are not administered through oral route, but still in vivo testing is required. And any modified release formulations like sustained release, controlled release, targeted release, colon targeted, gastro-retentive dosage forms, and timed release dosage form, pulsatile release, all these modified release dosage forms, definitely they are going to act systemically after absorption from the administration site. So that's why in vivo bioequivalence testing is required. And any fixed dose combination, we cannot give the exemption for the fixed dose combinations, like combination of two drugs, paracetamol with diclofenac sodium, or any cough combinations or cough suppressants. Many combination of drugs will be there in the single dosage form. In that case also establishing the bioequivalence by in vivo testing is required. And other category non-solution like suspensions, oral formulations, oral suspensions, oral emulsions, nasal, ocular, dermal, rectal, vaginal, all applications may be for the local acting or for the systemic absorption. Comparative clinical or pharmacodynamic studies are required by in vivo testing. And this is the slide when bioequivalence studies are necessary, but under other circumstances, like BE documentation is also needed to establish early and late clinical trial formulations. Like it is not a generic formulation, it is a innovator formulation, but you have done clinical trials long back and you are bringing the formulation into the market now. So recently fabricated batch versus previously conducted clinical trials batch need to be established. The correlation for that bioequivalence is required. And formulations used in clinical trials and stability studies, if they are different. In stability studies, you have used different formulation. In clinical trials, you have used different formulation. In that case, among these two, bioequivalence testing is required. And clinical trial formulations and to be marketed formulation, because we do the clinical trials with one batch, but we market the other batch. Between these two also, it is required to establish the bioequivalence. And other comparisons, like if you change the manufacturing process, if you change any new formulation, if you make any changes in the formulation, and before bringing that formulation to the market, we need to compare with the existing formulation. I mean, even after getting the approval, if you make any major changes in the formulation, if you change the excipient quantity or excipient you have changed, replaced with other excipient, manufacturing process you have changed, in that case also we need to prove the subsequent formulation is bioequivalent to the existing formulation. And the previously whatever we have discussed so far, in those all conditions we require to do in vivo bioequivalence testing. But this slide will give you some idea where we can do the in vitro testing itself. Just conducting the in vitro testing itself, it can be proven as bioequivalence. So under these circumstances, like any drug for which applicant provides the data to substantiate all of the following, it is nothing but is something related to BCS class 1 and BCS class 3. BCS is biopharmaceutics classification system. The highest dose strength of the drug, it may be 50 mg or 500 mg or 1000 mg, whatever the highest dose that is existed, that should be soluble in 250 ml of aqueous media at the range of 1 pH to 7.5 pH at 37 degrees centigrade. This is nothing but the definition of high soluble drug. If the drug is highly soluble and if the drug is highly permeable is nothing but at least 90% of administered oral dose should be absorbed on mass balance determination in comparison to the intravenous administered reference dose. If the drug is 90% absorbing and if the drug is highly solubilizing in 250 ml at the range of 1 to 7.5 pH, it is highly soluble, highly permeable, and at the same time, if 80% of the drug is dissolving in 3 medias, 0.1 normal HCl, 4.5 pH buffer, this is 0.1 normal HCl, 4.5 buffer, and 6.8 buffer, in these 3 medias at 100 rpm at the volume of 900 ml using apparatus 2 or 50 rpm using apparatus 1 at 37 degrees centigrade within 15 minutes if 80% of drug is releasing. So there are 3 criteria. One is it should be highly soluble, it should have high dissolution rate and highly permeable. In these 3 conditions, it will comes under the category of BCS class 1 or BCS class 3. In these cases, by conducting the in vitro testing itself, we can approach the FDA, approach the regulatory authority to get the approval of bioequivalence. And another category of drugs like different strengths. If you have 5 strengths, for all the 5 strengths, bioequivalence testing is not required. Only one strength can be tested, higher strength, and remaining strengths we can get the bio waiver by proving in vitro equivalence. Like the qualitatively, it should be having the same composition. I mean, all the 5 strengths, for example, we have 500 mg and 250 mg. 2 strengths we have. Innovator also is having 500 mg, 250 mg. These 2 strengths, 500 mg versus 500 mg, we will do the bioequivalence test. And 250 mg versus 250 mg, no need to do the bioequivalence test because your 500 mg and 250 mg, these 2 strengths are dose proportional and these 2 are having qualitatively same excipients and ratio of drug to the excipient is same. In that case, simply proving the dissolution profile in multimedia in 3 different medias like whatever I have shown in the previous slide in 0.1 normal HCl, 4.5 pH, 6.8 pH, if you prove and if your formulation is dose proportional, having same ratio of excipients and having same manufacturing procedure and having same composition, then in that case, we can get the exemption of bioequivalence testing with the lowest strength. So this is another criteria. Whenever we have different strengths, but they are all dose proportional or pseudo dose proportional, in that case, we can get the exemption of bioequivalence for the remaining strengths. Only higher strength we need to do the bioequivalence, remaining strengths we will get the exemption. These are the conditions where the bioequivalence testing is required. There is another criteria. In this case, bioequivalence testing itself, it is not required. Even dissolution testing also is not necessary. You might have got the idea where it is not required. If the dosage form is to be administered through parenteral route, like it is IV injection or intramuscular injection, subcutaneous injection or intraperitoneal injection, in that case, it is liquid dosage form directly administering into the systemic circulation. There is no absorption step. In fact, there is no absorption step. When there is no absorption step, there is no definition of bioavailability if there is no absorption. Because directly you are placing the dosage form into the systemic circulation. In that case, bioequivalence testing is not required. And if your drug is a solution for oral use, already it is in solution form. There is no dissolution step, only permeability. Permeability is the drug-related parameter. Your formulation drug and innovator formulation drug is same, but it is in solution form. No bioequivalence testing is required. If the new drug is a gas, when the new drug is a gas, again, there is no bioequivalence testing is required. If the drug is powder for reconstitution before administration, powder for reconstitution like dry syrups, dry suspensions. If the new drug is otic, ophthalmic, topical product prepared as aqueous solution, not suspension, again, bioequivalence testing is not required. If the drug is inhalational product or nasal spray, tested to be administered with or without any essentially same device as the reference product prepared as aqueous solution. It may not to be have the same different device, but still if it is aqueous solution administered through nasal route or as a nasal spray, then again that is not required. But provided in all these circumstances, under all these circumstances, the formula API present in your formulation and reference formulation should be same, because it should be chemically equivalent and it should be pharmaceutical equivalent. That should be remembered. Qualitatively, quantitatively, it should be same as that of innovator. Then only the bio waiver can be applicable here. I mean, bioequivalence testing is not necessary under these circumstances. So that should be pharmaceutical equivalent, chemical equivalent, and at the same time, qualitatively, quantitatively, your formulation should have the same composition as that of innovator formulation. Then only bioequivalence can be exempted. So here, now the introduction about the importance of bioequivalence testing and where the bioequivalence testing is required, where the in vitro testing is enough, and where in vivo testing is mandatory. I've already explained. So wherever the bioequivalence testing is required, under all those circumstances, chances of getting failure is more unless you have the practical biorelevant tool to predict the bioequivalence value. So where the IVIVC, in that case, establishing the correlation between in vitro testing and in vivo performance will come into the picture. I hope you got my point. Here, there are few dosage forms for which bioequivalence testing is not required, like injectable formulations, oral solutions, nasal solutions, and powder for reconstitution, because already they are in solution form, already they are administering into the systemic circulation directly. There is no testing required. And there are other category of dosage forms. If dose proportional lower strength, no testing is required. And there are few dosage forms, BCS class 1 drugs, high soluble, highly permeable, it is not required. But the first slide where I have shown several categories: narrow therapeutic window drugs, drugs to be absorbed through the GIT, drugs to be absorbed through the other part of site of administration, and high variable drugs. And in that case, in vivo bioequivalence test is mandatory. Wherever the in vivo bioequivalence test is mandatory, it can be either pharmacokinetic bioequivalence, pharmacodynamic bioequivalence, or clinical endpoint bioequivalence. We have three types of bioequivalence testing. One is pharmacokinetic, second is pharmacodynamic, third one is clinical. Clinical bioequivalence, pharmacodynamic bioequivalence, or pharmacokinetic bioequivalence. This testing is required though your formulation is generic formulation. You have already proven that you are chemically equivalent. You have already proven that you are pharmaceutical equivalent, but you also have to be proven that you are bioequivalent either by pharmacokinetic or dynamic or clinical endpoint. In this case, most of the formulations will fail because sometimes, we will do all formulation developmental trials and we match the dissolution profile in different media in the in vitro conditions. And among all the formulations, one formulation will be selected and it will be subjected for the bioequivalence testing. But while we may expect that we will get the value in between 80 to 125, because your dissolution profile, innovator dissolution profile, point to point, it is matching in all the medias. It may match. Dissolution profile is same as innovator in all the medias, and all other parameters. Disintegration time is same, hardness is same, friability is same, assay is same. You have used same excipients, same quantities, everything is same, but manufacturing process may be different. We don't know what kind of manufacturing process innovator have followed, and we are using different process. So, and API source, the innovator source of API, our source of API may be different. Particle size of innovator may be different. Particle size of your API may be different, and polymorphic behavior of innovator may be different. Your formulation may be different. These are all will not be considered. It will not be reflected in vitro testing, but will be reflected in vivo performance. That's why, though the formulation is passing in vitro, it may fail in vivo. And exactly where the IVIVC role will come into the picture. In that case, we need to establish, we should have a strong correlation between in vitro testing versus in vivo performance. And if you build that correlation and using this tool before conducting the bioequivalence testing, we can predict the bioequivalence value. Then we can proceed for the bioequivalence testing. And according to our prediction, we may get the similar values. But for that, in-depth knowledge on the product behavior in vivo and actual physiological conditions in vivo and actual appearance, actual behavior in vitro is required. Then only it is possible to build the correlation, because this is the core, it is the heart of the formulation development. Many formulation scientists will fail because of lack of knowledge on the IVIVC. So friends, because please focus from the next slide onwards, it is very important to understand how we can build the correlation between in vitro and in vivo correlation, in vitro and in vivo performance. So we have, as you all know, in vitro in vivo correlation. In short, it is been represented as IV, IVC. I in vitro, V is vitro, I is in, V is vitro, IV is in vivo, and C is the correlation. In vitro in vivo correlation we can build in 3 levels. One is level A, second is level B, third is level C. Just though you might already know about 3 levels of correlation, just as a quick understanding again, I'll give you a brief idea about level A, level B, level C correlations here. So everybody will do the dissolution in vitro, first of all, because the ultimate goal is to build the relationship between in vitro and in vivo. Here you can see this is level A correlation. How we can do the level A correlation, I will tell you here. Level A is nothing but we need to take in vitro and we need to take in vivo, both. In vitro is nothing but time versus percentage of drug release. Time versus percentage of drug release will give you an idea about a graph, how it behaves, whether it may follow 0 order release or first order release, but a profile you will get. Percentage of drug release versus time. I mean, time on x-axis, percentage of drug release on y-axis. Everybody will do this. This dissolution profile in different medias we need to do, first level. In different medias, you start from 0.1 normal HCl and then go up to 4.5 pH, then 6.8 pH, 7.2 pH, 7.5 pH. Because GIT, it has increasing order, pH of increasing in ascending order, starting from 1 pH to ending with 7.5 pH. In entire GIT, we don't know it from which part of the GIT the drug is going to absorb. And the conditions, I mean, prescribing conditions, is it to be taken on empty stomach or on fed state? We don't know. I mean, if you know, accordingly we have to decide the pH at which it is going to dissolve, the pH at which it is going to absorb. So that's why we need to perform the dissolution in various medias. The first level is we need to perform the dissolution in various medias with different RPM, with different, I mean, sometimes, you know, if it is sustained release dosage form, initial 2 hours it will be exposed to the HCl, I mean, in the stomach, then followed by it will be exposed to the alkaline pH in the intestine. Then these 2 medias are also should be covered in the dissolution profile. So first level is to get the dissolution profile in different medias with the different conditions. We can have 10 medias or 15 medias, and number, forget about it. As maximum as possible, try to generate the dissolution profile in several medias. Then from the literature, you can collect the information on plasma concentration versus time or serum concentration versus time. This will be available in the innovator formulation details, the plasma concentration at different time points versus time. Or else at least one bioequivalence testing if we conduct at pilot level, that information can be utilized. Plasma concentration versus time. Similarly, here percentage of drug release versus time as in vitro, and in vivo blood concentration versus time as another graph. You take this. This is available in the literature in the SBOA, summary basis of approval of the innovator. Because innovator formulation is already available, sometimes, you know, even in the patent also they may give this information, in vivo performance of innovator formulation, because already it is available. So now innovator formulation, in vivo performance, from this we need to do the deconvolution. Deconvolution is nothing but converting the plasma concentration into percent drug absorbed. There are several software tools are available at this moment, because giving all the details of how to convert serum concentration into amount of drug absorption is not possible, but you may know how to do this. Wagner-Nelson method, we have a standard method of converting plasma concentration into percentage of drug absorption. This is nothing but deconvolution. By Wagner-Nelson method, we can do all plasma concentration values or serum concentration values should be converted into amount of drug absorbed. Now time is constant. Here time versus amount of drug release. Here time versus amount of drug absorbed. In these two, now you remove the time factor, bring the amount of drug release here and amount of drug absorbed here. Now you take the graph by taking percent drug release on x-axis, percent drug absorbed on y-axis. Again, you try to build the correlation. Now here in this example, you got a straight line, but always it is not possible to get the straight line, you remember, because if we have done the dissolution profile, we have generated the dissolution data in several medias with several RPMs. All medias may not give you exact correlation, because let us say we have 10 dissolution profiles, but this is common. Percent drug absorbed is common. Only 1% of drug absorption we have, But percentage of drug release, different medias, different behavior. You try to build the correlation between any media, all the medias individually. First, percentage of drug release in 0.1 normal HCl versus percentage of drug absorption. Percentage of drug release in 4.5 versus percent drug absorption. Percentage of drug release in 6.8 versus percentage of drug absorption. With 50 RPM, with 100 RPM, with 75 RPM, with 500 ml media, with 900 ml, with 850 ml. In different several experiments we need to do, and our target is to build the correlation between in vitro performance versus in vivo performance. Percentage of drug release versus percentage of drug absorption. In different medias, every media you may not get the straight line, but in one media, in one RPM, in one volume of dissolution media, and definitely there is a chance of getting the correlation between in vitro drug release versus in vivo percentage of drug absorption. That media should be chosen as IV, IVC, and in that case, it is level A correlation. Sometimes in all the medias are also it is not possible to get the correlation. Then we need to move on to the level B correlation. Level B correlation, if the point to point matching, if it is not coming, then we need to move on to the level B correlation, where exactly we need to take mean dissolution time versus mean dissolution time in vivo. This is nothing but mean absorption time. This is statistical movement theory, non-compartmental pharmacokinetics. By using non-compartmental pharmacokinetics, we can have mean dissolution time on x-axis, mean absorption time on y-axis, and then again with different medias and mean dissolution time. Different media versus mean dissolution time. We can plot a graph, mean dissolution time versus mean absorption time of different dosage forms or different strengths or different time points. That is level B correlation. And if level B correlation also, if it is not possible, we have the other option. Anyhow, we have the percentage of drug release and you have the Cmax value, maximum plasma concentration. What is the percentage of drug release? What is the Cmax? Single point, directly single point correlation. Cmax value versus percentage of drug release. How much percentage of drug release within 8 hours and what is the Cmax? How much percentage of drug release within 4 hours? What is the Cmax? So like this, we need to correlate. This is level C correlation. So as an overview, we have level A, level B, level C. Of course, we have level D also. This is simple rank order process qualitatively. It is not useful for the regulatory purposes, level D. But level C, as I told you, it is a single point correlation, very less probability of getting the success using level C. And if you do the level B, this is statistical movement analysis, mean dissolution time versus mean residence time. And multiple level C also we have another option. It is a little bit useful and informative, at least 3 different time points covering different stages of dissolution profile. But the most informative and most useful is level A correlation in industrial point of view, a point-to-point correlation. If this is possible, then definitely there is a possibility of getting the success in predicting the bioequivalence. Now, as of now, I told you, if we have the in vivo plasma concentration data, then only either level A or level B or level C is possible. The question is here, it's very important. If anybody, any person from the industry, if you are attending this session, you may understand in a better way. Most of the times we may not have the plasma concentration values in our hand. In vivo data, if it is not available, we have never done any bioequivalence testing and we don't have the innovator information. We have only the dissolution profile of test product. We have the dissolution profile of innovator product. Only dissolution data is available. If only the dissolution profile is available, then what is the solution? How can we build the in vitro in vivo correlation? How can we predict the bioequivalence? This is the question. If we don't have the plasma concentration, and then definitely there is no possibility of knowing about percentage of drug absorption. We don't know mean dissolution, mean absorption time. We don't know mean residence time. We don't know the Cmax value, exactly what is the Cmax value of innovator. We don't know anything about the innovator formulation. And we never done any bioequivalence testing. Can we predict the bioequivalence? Yes. Still, it is possible to predict the bioequivalence. That I will explain you in the case study. So let me begin with the case study. This is more important part of my presentation today. This is actually practical problem what we have faced in our industry. We have done, in industry generally, once the lab scale development is done, once the formulation development at laboratory level, if it is completed, then it will be transferred into the technology site, where actual manufacturing site, GMP site. We need to do some scale up studies. Then once the scale up is completed, once we optimize the process parameters, then finally we need to do the exhibit batches. Exhibit batch is nothing but the submission batches for the approval. And using these the 3 exhibit batches are required with final commercial scale size. At least minimum 1 lakh is the batch size is expected by the FDA. So once the like similarly like this 3 exhibit batches are required. For 3 batches, bioequivalence testing is not required. Only 1 batch it can be tested for the bioequivalence against the innovator formulation. And with this 1 batch, if we prove our formulation is equivalent to the innovator, then remaining 3 batches are also accepted. Subsequently, all commercial batches are without conducting bioequivalence testing, we can release them into the market. So this is the procedure actually. So generally, the required number of subjects are also 18 subjects, minimum number of subjects, healthy volunteers are required to prove our formulation is equal to the innovator. So what we have done, this case study, if I explain the statement of problem, you will understand what was our problem and how we could able to resolve by using IVIVC. So we have done one pilot there are 2 types of studies, pilot and pivotal. Pilot is nothing but using less number of subjects to get the confidence before going for the pivotal. Pivotal is actual bioequivalence testing. Pilot is small scale. By using the scale up batch also, a pilot study can be done. And then by using the exhibit batch, we can do the pivotal study. Because directly if we do in the large number of population, if we get the negative result, then the cost of the bioequivalence is very high. It will go into the vein. So that's why generally every industry will do the first pilot study in less number of subjects. They will get some confidence. What is the exact value of Cmax and exact value of AUC we are getting. That once the confidence is coming, then they will go for the exhibit batches and pivotal testing in large number of population. So what we did earlier, a pilot bioequivalence study of an exhibit batch. Directly we fabricated 3 exhibit batches, 3 batches with 1 lakh commercial batch size. That is extended release formulation with a 12 hours release profile. It was evaluated in 14 volunteers first. Directly, actual number of volunteers required for our study was around 45 subjects. 45 number of subjects was required. And directly, if we do in 45 number of subjects, the cost of the study, it was going behind 1 crore. So our sponsor have decided, in spite of going for the 45 number of subjects directly, let us test it in just small number of volunteers, 14. Then that too, we require to do 2 studies. Like one is fasting state, other one is fed state. Fasting is nothing but on fasting we need to do. Fed is after taking the breakfast, we need to administer the dosage form, either breakfast or the lunch. That volume of meal, how much have to be taken, also has to be. It is prescribed by the FDA. So 2 studies are required, fasting and fed. Among these 2 studies, fed study is more critical. If we get the fed study pass, if we pass in the fed study, definitely we can expect that fasting study also definitely it may pass. So as per the drug physicochemical properties and other characteristic features, we concluded that fed state is more critical. And directly our sponsor have suggested to do only testing in 14 volunteers initial level. We did the 14 volunteers testing in fed state. You can read here. And the outcome, it met the acceptance criteria for all the parameters. Outcome means the total Cmax value and AUC ratio. Here it is called as T by R ratio, tested to the reference ratio. T by R ratio, it should be, as I told you, 80 to 125 for the Cmax, 80 to 125 for the AUC also. So now in these 14 volunteers in fed state, when we have done the study, all the results were met the criteria. 105 T by R ratio, we got it, and 100 T by R ratio for the AUC, we got it. So what we feel, as 14 volunteers have shown 105, because 105 is in between 80 to 125. Based on the pilot results, using the same test formulation, using the same reference formulation, we have conducted the pivotal study. Pivotal study, as I told you, requires large number of subjects for the submission purpose. So there, the test product failed to meet the bioequivalence criteria for Cmax, both in fed condition as well as fasting condition. Now it was surprise because the same formulation of test and same formulation of the reference have given positive results, positive outcome in 14 volunteers. When the number of volunteers were increased, the result was not meeting the acceptance criteria. It was supra bio results. We got it. Supra means higher than 125. It should be between 80 to 125. We got 133, something like 133 or 134. It was to our surprise. Earlier it was 105. Now it has gone to 133. So now there is a need to find out the root cause of the failure of pivotal studies. Why it is failed in pivotal? Why it is passed in pilot? By building in vitro, in vivo correlation, and finally deciding way forward for the success of pivotal bioequivalence study. So this case study, as a statement of problem, I will try to explain how we have built the correlation by using the dissolution profile alone and by using IVIVC alone separately. As I told in previous slides, level A correlation only we have built. How we have built the IVIVC in using plasma data of this failure, and how we have brought a conclusion, how we have drawn a conclusion based on the dissolution profile alone. You can see here the data obtained in pilot as well as in the fed. I have represented in a table and followed by a graph in the down. Are the pilot and pivotal studies are conducted properly, or is there anything went wrong? Because in the pilot we got positive result and in the pivotal we got negative result. Is there any scope to use the same exhibit batch for the repeat testing? If we repeat the testing again, is there any chance of getting the positive result? That was our question, first of all. So here the Cmax values, because the formulation failed in Cmax only. AUC was good. AUC, it was in between 80 to 125, both in pilot as well as in pivotal. But Cmax value, it failed in pivotal, but it passed in the pilot. In pilot, as I told you, we have done, remember, we have done only FED study in pilot, but we have done both pilot and FED and fasting in pivotal. So here pivotal fasting and pivotal FED and fasting in pivotal, and pilot FED only. So these three Cmax values of test, three Cmax values of reference. So it takes more time to grasp and to understand very critically. But only as a just I'll tell you as a main crux. Here you see the difference between Cmax test versus reference. The difference between these two, 487. And even in the fasting also, FED and fasting both, Cmax between these two, it is 485. 485, 487. The difference between Cmax value, it is not the ratio I'm telling, it is the Cmax value difference is 487, 485. And even if you look into the data, FED minus fasting, the difference is 274. Cmax FED minus pilot FED. Pivotal FED minus pilot FED, it is 144. And reference in case it is 484. 484, 484, here it is matching reference. Reference is behaving similarly, but the test formulation here 148, 148. But pilot versus pivotal, if you see, FED only 148. Here the difference, here the difference is 487. The same test formulation, when it is repeated, why it has behaved differently in a small number of valencies and large number of valencies, then the systematic approach we have done completely individual subjects values when we have plotted as a graph, T by R ratios, frequency distribution of T by R ratios of individual 14 subjects. Now we can look into the 14 subjects individual T by R ratios. One subject is 74, another subject is 80, another subject is 81, and three subjects are just around 100. Three subjects are 100, and remaining 8 subjects are above 100, and at least few subjects 173, 129, 179 are also there. Now these are the results of 14 valencies. In the first 14 valencies, these yellow colored 3 valencies, these 3 valencies, actual frequency distribution plot, if you see, it is skewing towards right side because frequency 100 is in the middle, 80 is in the left side, 125 in the right side. If you see, more number of subjects are coming towards right side of the graph, not to the left side. Left side there are only 3 members, but right side there are 8 members. Sorry, not 8 members. How many are there? 8 members. 8 members are right side, 3 members are left side, 3 members are in the middle. And if you have the graph frequency normal distribution skewing in both the sides, center point in the middle, bell-shaped curve, if you get, you will get the average value accurately. But only 3 subjects are lower side and 8 subjects are higher side. These 3 subjects, lower side subjects, they have misleaded our result to have the 105 Cmax value. That they are the culprits to get the lower side Cmax value. If they are not there, if these 3 members are not there, and then definitely we would have got higher Cmax value in pilot also. That was our conclusion. I'm repeating once again, in out of 14 valencies, if all the valencies behave similarly, you would have got 133 T by R ratio in pilot also. In pilot study, there are 3 subjects, they are like outliers. There are 3 subjects have shown completely different response than the others. These 3 subjects led to the misleading the results completely. And we thought because of these 3 subjects, this statistical analysis we have not done before proceeding for the pivotal. And if we had done this statistical distribution, statistical comparison understanding, then we would have not gone for the pivotal. So because of these three subjects misled us. These three subjects were the culprits of getting the 105 as the average TBR ratio. So accordingly, we have increased the number of subjects, and when we have repeated, we got 133. Because upon repetition, this kind of performance was not there in 45 number of subjects. These three are the culprits. Because of these three people, overall total Cmax value have come down. So for this, again, we have done, and we wanted to do some correlation. We wanted to build the kinetics. We wanted to do the correlation between in vitro and in vivo correlation. If current formulation is not suitable for the repeat bioequivalence studies, then what is the correlation to predict the bioequivalence? Then we thought of conducting the in vitro in vivo correlation. Now here this graph, just I told without having in vivo data also, simply from the dissolution also we can get the there is a chance of getting the correlation with in vivo performance. Here this is the dissolution graph in 0.1 normal HCl. In 0.1 normal HCl, dissolution of innovator, dissolution of test. This is reference. Blue color is reference and red color is test. You can see here higher side, test formulation is higher side. That itself it is indicating your test formulation is faster than the innovator. And when we have calculated the this is the equation from the log linear equation, 24.976 linear x plus 51.282 in case of reference, and 28.413 in case of test. 28 versus 24, this 28 divided by 24, if you do, the ratio is 114. That itself it is higher side. And similarly, another media, this is another media 4.5 pH. 4.5 pH, this is both zero order as well as first order, both it is following, because R square value will give you the idea whether it is following zero order kinetics or first order kinetics. This linear is nothing but linear also 0.99, linear also 0.95. That means, and in case of log linear 0.95, 0.99. Both it is following zero order, combination of zero order as well as first order. Because if R square value is more than 0.9, we can conclude it is following that relationship. So here, if you see the ratio in both slope ratios in both the test as well as reference in zero order and first order, the actual value is 138 and 133 from the dissolution only. This is also from the dissolution. From dissolution data, just slope value, ratio of the slope values of test versus reference, T by R ratio is actually 138 and 133. Now, if you look into the overall data, not the overall data, this is another type of correlation from the IVIVs. As I told you, 0.1 normal HCl, percentage of drug release versus percentage of drug absorbed we have taken. Percentage of drug release we have taken on x-axis. Percentage of drug absorbed we have taken on y-axis. This is in 0.1 normal HCl, 0.01 normal HCl for the reference, and this is for the test. And this is 4.5 pH for the reference, 4.5 pH for the test formulation. Here also we have different slope values. And from all these T-bar ratios, calculated T-bar ratio from in vitro, as I told you previously, 114 in 0.1 normal HCl and 133 in 4.5. And IVIVC of all subjects, 53, 59. IVIVC, if you remove those three subjects, 116 and 132. And actual values what we got, 118 in fasting and 130 in the fed. Now in the summarized table, you can see from the dissolution also, our prediction, if we had done the prediction before proceeding for the pivotal, 114 is our prediction, actual value 118. 133 is our prediction, actual value is 130. So now see the difference. Just 4% difference between in vitro and actual. In vitro and actual, 3% in the fed, 4% in the fasting. How closer we are? The total range allowed is 80 to 125, but here actual prediction 114, actual value is 118. Prediction 133, actual value 130. In case of IVIVC, in case of dissolution, this is the value. In case of IVIVC, considering all subjects, 53, 59. That means there only it went wrong. If we have removed the three subjects, then 116, 132. 116 is very close to 118. 132 is very close to 130. Then removing the three subjects, and if you do the bioequivalence prediction, then we would have not gone for the pivotal study. We would have not gone for the pivotal study. Then subsequently, we have made some changes in the formulation. We have made few changes in the formulation, and again, we did the IVIVC, we did the dissolution kinetics, both alone dissolution kinetics and IVIVC in 0.1 normal HCl and 4.5 pH. When we did the IVIVC and dissolution kinetics, prediction 100, it was in 0.01 normal HCl, and 1 or 2, it was pH 4.5 pH buffer. and same results we got it in the actual pivotal bioequivalence study. Of course, 3 more bio exhibit batches we have fabricated, and we got 100 and 1 or 2. The same 100% is our accuracy in prediction. So that's why we have reduced the number of batches. Again, if this tooling had been not there, then how many number of batches we would have been fabricated, you can imagine. In that way, just by making only one batch, we have got the success in getting the bioequivalence. So this is how the in vitro tooling or in vivo IVIVC will helps us to reduce the number of batches and to get the bio success. Moving on to the case 2, second case with respect to the bio waiver directly. This is the another formulation. It is earlier, as per the guidance, clinical endpoint bio study was required. Clinical endpoint bio study, clinical, that means the testing should be done in the patients. That formulation is for the, it is to treat the ulcer. It is for the ulcer treatment. Ulcer patients we need to select, and we need to do the clinical endpoint testing by taking test formulation in one group and administering reference formulation in one group, administering placebo formulation in another group. That was the guidance. Testing in the patients is unethical. So we approached the FDA to give the bio waiver by simply conducting the in vitro studies. These following studies have been proposed by our side. Then the FDA have accepted and given the clearance, and they have changed the guidance from clinical endpoint bio study to the in vitro studies. So now onwards, any formula for this particular formulation, testing in the patients is not required. Just testing in vitro is sufficient enough. In that, there are 3 levels of testing. First of all, as I told you, test and reference formulation should have the same API, and they should have qualitatively, quantitatively same excipients except flavor and color, and they should have physicochemical characteristics comparable. And finally, acceptable bioassay of the test and RLD formulations, like API sameness. Established API similarity can be shown by API composition and FTIR spectra, UV spectra, solid state 27 aluminum NMR, differential scanning calorimetry, TGA, and powder X-ray diffraction. By getting the data by all these techniques, we have to prove that our API, innovator API, is safe, including the particle size. And in vitro, we have to check the pH of our formulation and reference formulation, specific gravity, viscosity, change in apparent viscosity with addition of acid, because it is suspension, redispersibility time, sedimentation rate, and comparative acid neutralizing capacity, comparative release at pH 1.2. All these are the in vitro testings are need to be done as part of establishing the pharmaceutical equivalence. And as part of bioequivalence, clinical endpoint bio is been replaced by in vitro equilibrium testing as bioassay. That was in vitro equilibrium binding study with human serum albumin or bovine serum albumin, in vitro equilibrium binding study with bile salts, in vitro kinetic binding study with the bile salts, in vitro enzyme activity study by binding with the pepsin. These are all the in vitro bioassay tests that have been proposed by us, and if they have accepted in this, if we prove our formulation is equal in all these tests, both in API, in vitro formulation, and bioassay, then we can conclude that our formulation is equivalent to the innovator formulation. In that way, we have succeeded, and we have proven that our formulation is equivalent to the innovator, and we have submitted our application form as an ANDA application to the FDA, so that we have get the waiver of clinical endpoint biotesting, because getting the testing in the patients is very critical job, and even it is unethical. So we have approached the FDA by proposing these alternative in vitro equivalence testings, and FDA also have accepted. So here, with this brief understanding on these 2 case studies, I would like to conclude with the role of formulation scientist in getting the bio success. We should have the thorough understanding of the API knowledge, because API means if it is paracetamol itself, it is not sufficient. As I told you, we should have the IR spectra, UV spectra, HPLC spectra, and DSC, XRD, and all other possible techniques we should see, and polymorphic behavior, hydrophobicity, and dynamic vapor sorption, and particle size, especially solubility, partition coefficient of all our API and API from the innovator we need to extract, and that we need to check. Thorough understanding of the API is the first step. Thorough understanding of the innovator product, how it is behaving physically, how it is behaving chemically. By means of reverse engineering, we can do all sort of testing. That is second level. And then we need to match the dissolution profile in multimedia, and where in multimedia similarity factor, we can use F2. And as I told you, dissolution kinetics, we can do the 0 order release or first order release kinetics. We can consider the slope value, and by making the slope ratio of test formulation by the reference formulation, we can have some certain idea of bioequivalence, bio value, T-bar ratio in actual in vivo testing. And then if we have the plasma concentration values of innovator, we can establish the IV-IVC by deconvolution technique by taking percentage of release on x-axis, percentage of absorbed on y-axis. That will give you another idea of predicting the T/R ratio. Then we can finally predict the BE results, IV-IVC and modeling kinetics. Then getting the success rate will increase. Then you can become a successful formulation scientist in formulation research and development. So with that conclusion, I would like to wind up the today's presentation. So this is our team from New Height research team, both formulation and analytical team. And I'm really thankful to work for the New Height Pharma, where I got very much tremendous knowledge, because I'm working under a legend, pharma legend, Dr. V. Venkateshwarlu sir. He is my guru in my B Farm, and fortunately, I got another opportunity to work under him and to gain more knowledge and to update our knowledge every day. So he's expert in the bio. So under his guidance, we all are working. And thank you very much to him once again, because he gave me this opportunity, accepted my invitation, my request to share my knowledge with you all people, because two practical case studies I have used in the presentation. And thank you very much. Once again, I am thankful to the Pulla Reddy College team management and the faculty members, especially Madhav Reddy sir and Dr. Prashanti, who was my junior in my M.Pharm. She requested me to share my knowledge in this presentation. And thank you very much. Thank you once again. And if you have any queries in with respect to IV-IVC or bio-waiver or bioequivalence testing, you can text in the text column, so that if it is possible from my end, I will try my level best to answer all of your queries. If not, I will get back to you later. Thank you very much. Nasheeb? Hello? Nasheeb, are you there?
Yes, I think there's some technical problem. So Mr. Nasib Pasha isn't joined. Thank you, Mr. Vijay Kumar. We'll just go into the questionnaire session now.
Mm-hmm.
So we have some questions from the audience.
Okay.
With regard to, initially there was a question with regard to the dry powder inhalation formulations. Do we need to do this IV-IVC correlations?
No, actually, dry powder, that is what I have already mentioned in the slides. Dry powder inhalations, if it is for systemic absorption, we need to do the clinical endpoint bioequivalence testing based on the requirement as per the guidance. If it is only oral solution form or directly gaseous form, and then it is exempted. Okay. And powder for reconstitution, powder for inhalations, for systemic absorption, definitely we need to do as per the guidance requirements.
Okay. And there is one more question from Shilpa. Is it possible to evaluate bioequivalence of a test, that is the immediate release and the modified release? Can we compare between an immediate release formulation and a modified release formulation?
Yeah, I got our question, Shilpa question. Actually, preliminary requirement, the foremost requirement to conduct the bioequivalence testing is our test formulation should be chemically equivalent and pharmaceutically equivalent. And I've already defined clearly, chemical equivalence is nothing but our API and dose should be same. Pharmaceutical equivalence means our formulation and the test reference formulation should be same dosage form and same route of administration. If innovator formulation is immediate release, our formulation is modified release, we cannot compare these two formulations as bioequivalent, because in this case bioavailability testing will come into the picture rather than bioequivalence. Our formulation behavior is different, innovator's formulation is different. If you do any modified release dosage form, it comes under a separate category 505(b)(2). It is a hybrid NDA. It is not ANDA completely, because it is a new dosage form. Whenever it is a new dosage form, new strength, new route of administration, new fixed dose combinations, it is not possible to match the result, match the bioequivalence with that of innovator, because innovator formulation is immediate release formulation, is frequency of administration daily 3. But your frequency of administration, maybe you want to come up with one single day dosage form. modified release dosage form. And if you conduct the bioequivalence testing, you will get the failure, because the rate of absorption and extent of absorption from your formulation is different, their formulation is different. So rather than bioequivalence, generally bioavailability testing is required, and this will comes under 505(b)(2). It is a separate application form. It is neither NDA nor ANDA. It is hybrid NDA and ANDA. That is separate session, and it requires other additional studies are also. Sometimes FDA expects to do some clinical testing also, in addition to the bioavailability study. It all depends on the type of molecule used. Yeah.
Okay. We also have a session tomorrow, that is on this 505. So I think their doubt will be better clarified in that session. And there is one more question from Kushbu. So in this, they have given a doubt in your example 5. That is where you have given a different strength of the dosage form. The higher strength bioequivalence is performed, whereas for the remaining strengths, it was given biowaiver. And the F2, the similarity factor, could match with the bioequivalent strengths in all media. So with regard to this, she has just asked.
Yeah, I mean, that's it. That's it, the question?
Yes, that is the question.
Is there any continuation to the question?
No.
It's like a statement. Answer itself. Answer is given already in the question itself. For example, if we have 5 strengths and all the 5 strengths are dose proportional or pseudo dose proportional, there is 2 difference. Dose proportional is nothing but not exactly proportional quantity of blend will be used for the compression. Pseudo dose proportional is nothing but we'll be having the same weight tablet to tablet. For example, one strength is 200 mg, one strength total weight is 200 mg, another strength total weight is also 200 mg, but the dose is very less, 200 micrograms in one, and another one is having 100 micrograms. So except the drug, remaining excipients, everything is same. In that case, it is called a pseudo dose proportional. So if our formulation is pseudo dose proportional or dose proportional, only biotesting will be done for the higher strength, and sometimes if it is a safety concern, if it is there, we will do with the lower strength also. So either higher strength or lower strength, one dose, one strength only will be subjected for bioequivalence, and remaining all the strengths should be similar in dissolution profile in all the media as that of your biostrength, not with the innovator. Now, no need to compare with the innovator. Our formulation, one higher strength formulation dissolution profile in all the media versus remaining strengths dissolution profile, it should be same. F2 value should be same. I mean, F2 value should be higher than 50. F2 is the similarity factor. There is a formula to calculate similarity factor. It should be more than 50. If we get more than 50 in all the media suggested by the FDA, then that itself it is sufficient enough to get the bio waiver for the remaining. But we should have the same composition, same drug to excipients ratio, dose proportionality, and same manufacturing procedure. And remaining all other quality attributes are also should be same. Then only it should be subject. I mean, we can get the bio waiver for the remaining strengths.
Yes, sir. And there is one more question from Mr. Madhusudan. So he wants to know the therapy category of drug which is used for clinical endpoint. In your case studies, what you have discussed, he just wants to know the therapeutic category of the drug which was used.
Rather than therapeutic category, in other way I can answer this question. Actually, it is not based on therapeutic category, based on the site of action, like locally acting drugs. If our formulation is locally acting in the GIT, only it is not undergoing any absorption. For example, as I told you in the BioWaiver clinical endpoint, one case study, it is for the ulcer treatment. Ulcer, it may be in the lower part, in the upper part. Sometimes colonic ulcer also it will be there. Sometimes gastric ulcer. So our formulation is subjected for the treatment of ulcer. It will not undergo any absorption. If the drug is not undergoing absorption, but still if you want to do any pharmacokinetic endpoint study, because the drug itself, it is not coming into the blood. If you do the pharmacokinetic study by collecting the blood sample or urinary sample, and more variability. Absorption absence means at least definitely sometimes 1 to 2% of drug there is a chance to absorb. And if you want to estimate that drug in the plasma, and if you want to compare that drug in the plasma between test and reference, definitely more error will be there, because your
Yes, there's some technical problem. Just for 2 minutes, participants.
Yeah, sir will join soon from sir side. Yes, sir came.
Point. It is not with respect to the category of therapeutic category of the drugs. if you ask me therapeutic category, I can say ulcer, anti-ulcer, and colonic cancer, colorectal cancer. So that kind of molecules will come in. Many skin related fungal infectious diseases, topical infectious diseases also can be placed under those category. But it is
Some technical problem from the speaker side. He will join soon. Prashanti madam, are you audible?
Yes, I'm audible.
Yeah. Speaker will join soon. And meanwhile, we'll have a few announcements regarding assessments come feedback. Please visit G. Pullardi Kalya Pharmacy Hyderabad YouTube channel. For assessment come feedback, please visit www.gprcp.ac.in. Subscribe to GPRCP Hyderabad YouTube channel for more updates. Assessment come feedback link will be activated after 1 hour of the session and it will be active for 24 hours only. And tomorrow, that is day 6, 13th of July 2020, the first session will be at 10:30 a.m. onwards on topic Overview and Scope of Drug Regulatory Affairs by speaker Dr. Santosh Kumar Narla, NAG Manager US Regulatory Affairs, Dr. Reddy's Laboratories Limited, Hyderabad. And session 2, that is tomorrow, day 6th of 13th July. Second session starts from the 3:30 p.m. onwards, which on the topic which abbreviated approval pathway to choose for a drug product 505(b)(2) or ANDA by speaker Dr. Venkateshwari Muthukrishnan, Associate Director, Nuventra Pharma Sciences, Durban, North Carolina, USA. For assessment come feedback after 1 hour of this session, it will be activated. So we are waiting from the speaker side. Due to technical issue, he couldn't join.
hello.
Yeah, speaker has joined. Yes, sir. Sorry for the interruption. Technical problem.
Sorry, actually.
Yes, sir. Yes, sir.
Okay. Shall I start? I mean, share screening is not
Yes,
required
sir.
no, at this moment now.
It's not required, sir. So
Screen
I'm
sharing is
highlighting
not required, right?
Yes, sir. I'm highlighting few
Okay.
questions. Please answer to those questions.
Okay.
Uh,
Sure.
Yes, sir. Is it possible to evaluate bioequivalence of test immediate release and test of medium release formulation,
Yeah,
modified
that I've
release
already
formulation
answered.
versus?
This I think.
Yes, sir. Yes,
This
sir.
I've already
One,
answered. I mean, this.
and one more question. Sunny,
Yeah, this
in
is
your
also from
example,
Kushbu Bharat.
yes, sir. Did you cover?
5
And
strengths of
third
higher
one,
strength BE is performed. Already I've covered this question. I've
yes,
answered.
sir. Yes, sir.
Yeah,
One more
this one only
question.
actually I was giving the explanation, but how far?
Therapeutic category of drug used
I mean,
for
may I know the
clinical.
therapeutic category of drugs used for clinical
Yes, sir. And one more question.
endpoint?
One
So
more
the
question by
while
Veeresh
give.
Shetty, sir. Yes, sir. Is there any biowaiver for brain-targeted drug delivery system through nasal route?
It's not based on the targeting to which part of the brain, to which part of the body the biowaiver is being given. It's, as I told you, based on your dosage form design, what kind of formulation it is. If it is when brain targeting, nothing but it is advanced drug delivery system, and definitely biowaiver is not at all possible. And in fact, bioequivalence also it may be not possible, because unless we have the reference product in the market, approved product in the market, and as the question itself, it is indicating brain targeted delivery. We have very few brain targeted delivery systems through nasal route are available, approved in the market. So unless we have that kind of formulation in the market, we cannot expect the comes under 505(b)(2) new formulation designs everywhere. It comes under 505(b)(2) to do bioavailability studies and other certain toxicological studies to get the approval.
Yes, sir. One more.
Yeah.
Do we have to do bioavailability study for nano formulation given by IV route?
No, IV route, any formulation, either it may be nano or micro or any conventional formulation, if it is given by the IV route generally it is a solution form. Simply, if we match the Q1, Q2, Q1 is nothing but qualitative composition, Q2 is quantitative composition. If Q1, Q2 is same, then no need to do any bioavailability or bioequivalence kind of thing, because bioavailability definition itself, it is indicating rate and extent of the drug that undergoes systemic circulation, that reaches systemic circulation. Here in IV route, there is no absorption step. Whatever the drug directly we are in formulation, we are injecting, everything it comes into the systemic circulation. There is no point of, no bioavailability or bioequivalence requirement in IV formulations.
Okay. And one more, sir.
Any other?
Yes, sir. Yes, sir.
Pratibha Paoluri, I said BCS class 2, 3, 4 products can be also biowaivered. No, for BCS class 1 and class 3, as they are highly soluble drugs, there is a biowaiver applicability. But BCS class 2 and class 4, they are suffering from BCS class 2, especially having low solubility, class 4 low solubility as well as low permeability. And we can never expect the biowaiver, though you have excellent IV IVC also. So it is not at all possible to get the biowaiver for class 2 and class 4. In certain cases, class 3, providing other justification like matching the dissolution profile in all the media and having the IV-IVC tooling, we can get the biowaiver. There is a chance for the class 3, but not for the class 2 and class 4. Yeah, next question, please. Are there any limitations to IV-IVC? Why not? Definitely, because every advantage, definitely it will have its own limitations besides having so many advantages. So similarly, IV-IVC also having the limitations, because it is very difficult to get the IV-IVC in most of the formulations. Because all the formulations, as I told you, it is not possible to get the IV-IVC, because sometimes you may have the dissolution profile, immediate release formulation, everything will, because if it is BCS class 1 or BCS class 3, whatever the
Sir.
Hello, Basha, voice not coming.
Madam.
Yes, madam, please carry on.
No, the speaker voice is not coming.
Oh.
Yes, madam.
Yes, madam.
Sir has joined again.
Okay.
So some internet problem here. Sorry.
Yes, sir. We were there in the last question. Are there any limitations to IVC?
So that is what I was explaining. Besides
yes.
having advantages, there are so many limitations. Similarly, all dosage forms, it is not possible to get the IVC by either level A or level B. Sometimes level C also may be not possible. So that is only the limitation.
Okay.
Yes, sir.
Other than these, we have only the appreciations from the participants. So we move on to the tank note as there are no more questions. And if any participants want to further ask questions, you can also mail to the speaker. We will be providing the mail ID in the brochure.
Yeah, that's better, I think. If you have any further questions,
sir.
definitely I will ready to share responses.
Yes, sir. So myself, I'm going to propose the tank note. Myself, Dr. D. Prashanti, Associate Professor on behalf of G. Reddy College of Pharmacy and participants of e-FDP on research updates in pharmaceutical formulations and analytical method development program organized in association with IPA Telangana State Branch would like to express appreciation for your presentation, sir, on the role vitro in bioequivalence studies and vivo. Sir, you have highlighted the importance of IVC correlations, and they have started it from the basic definitions of bioavailability, bioequivalence, the therapeutic equivalence, and the chemical equivalence, the different levels in IVC correlations, when bioequivalence studies are required, when biowaivers can be obtained, the category forms where bioequivalence studies are not required, and we have also discussed the role of the formulation scientist. The case studies which are being presented, it shows your contribution in years of research and also your depth of understanding. And the session was really informative, and I can also assure the participants who are majorly faculty members can incorporate in the research in the coming years. That is with regard to without the in vivo studies, how we can proceed for the IVC correlations and the other methods. And there are more appreciations from the live YouTube chat, which I would like to share, sir. That is, the session was knowledgeable, it was informative, it was nice and excellent session. I can also assure the expectations of the participants from this topic have been met, and they have gained some knowledge from this presentation, and also they have cleared their understanding of the importance of the IVC correlations in today's generic market, where we have the generic, which is the pharmacy market, which is at present in the world. And I thank you, sir, for taking the time to speak and accept our invitation in spite of your busy schedule. I once again thank you, sir, on behalf of our conveners, Dr. B. Reddy, sir, Principal of G. Reddy College of Pharmacy, and Dr. B. Prabhashankar, President of IPA Telangana State Branch, the organizing secretaries, Dr. Y. Padmavati, Professor in Department of Pharmaceutical Analysis, and Dr. K. Lata, Professor in Department of Pharmaceutics. And on behalf of the organizing committee, Mr. P. Ravikumar, Mr. Sheikh Nasir Basha, Mrs. K. Pallavi, Mr. N. Raghavendra Babu. Once again, I thank all the participants also for their patience and interest in attending, as today is Sunday and they have also cooperated with us. Thank you to all the participants also, and thank you, Mr. Vijay Kumar, sir. Thank you for accepting our invitation and giving us this nice presentation. Thank you. And
thank you, madam. Thank you. It's my pleasure to be part of this wonderful program. Thank you. Thank you. Thank you, all of you. Thank you, all the faculty members, for your cooperation and patience listening. Signing off. Bye.
Yes, sir. Participants, there is an announcement. So regarding your assessment and feedback, please visit college website www.gprcp.ac.in. Assessment and feedback link will be activated after 1 hour of the session, and it will be active for 24 hours only. Subscribe to GPRCP Hyderabad YouTube channel for more updates. If any participants having any queries, feel free to drop a mail to seminars gprcp.ac.in or visit our website www.gprcp.ac.in. And tomorrow, that is date is 13th July 2020. We have two sessions. First session at 10:30 a.m. onwards. Topic overview and scope of drug regulatory affairs by speaker Dr. Santosh Kumar Narla, NAG Manager, US Regulatory Affairs, Dr. Reddy's Laboratories, Hyderabad. Session 2 at 3:30 p.m. and topic which abbreviated approval pathway to choose for a drug product 505(b)(2) or ANDA by speaker Venkat Ishwari Muthukrishnan, Associate Director, Nuventra Pharma Sciences, Durham, North Carolina, USA. On behalf of G. Pulla Pharmacy, I thank conveners and organizing secretaries and organizing committee members, and special thanks to today's speaker Dr. N. Vijay Kumar, and thanks to everyone for joining us. We appreciate you being here. We will see you tomorrow, that is day 6 of e-FDP. Thank you, and signing off today, your Sheikh Nasib Basha. Thank you.